Welcome to our discussion on membrane potential. And this is one of the more abstract and tricky concepts for students in introductory physiology. It's actually this particular topic that made me start to wonder if recording these videos so students could re-watch would actually be helpful. You know, however many times I presented in class, maybe going over it just a couple more times on your own from the same person in the same type of language is going to be helpful. So hopefully this helps get the point across so we can build on it later, because this is an important concept for when you move on to that next step in physiology, moving on to neurophys, cardiovascular physiology. Those are going to be core concepts for how a neuron and some of those other cells function. When we think about what a potential is, let's forget about the cell for a moment, but when we consider what a potential is in physics, it's going to be described as a separation of charges. So if we think about a battery, for example, we had this separation of charges inside the battery. You connect the terminals, and that separation is going to wind up creating a current through that new wire. Likewise, if we connected the internal and extracellular surfaces of that membrane, as if we were connecting the terminals on a battery, we would see a voltage measured across that battery, or a potential. When we're trying to figure out exactly how this occurs, we have to start wondering what charges are going to be involved, where are they, and how do they actually create this current. So what I tell students first off is think about a salty banana. That starts getting us thinking in terms that we already know. We already know that salt is sodium chloride and a banana is high in potassium. So if we look at those particular ions, we see where they reside in a normal living cell. We see a high potassium concentration inside the cell. We see a high concentration of sodium and chloride, or salt, outside the cell. This particular imbalance of sodium and potassium is going to be maintained by the ongoing actions of our sodium-potassium pump. Every time we break down an ATP, we pump 3 sodiums out of the cell, we pump 2 potassiums in. So as long as that pump is working, we will maintain our imbalance of sodium and potassium on opposite sides of that cell membrane. Now, of course, those aren't the only ions directly involved. We also see a lot of negatively charged amino acids hanging out inside the cell. And we also see calcium, a positively charged ion, outside the cell. So these ions, combined with our sodium, potassium, and chloride, are the major players for determining the membrane potential of a cell. So here's the situation that we've just described. We have a cell with a working sodium-potassium pump, always pumping sodiums out and potassiums in. So we get a high concentration of sodium outside the cell, high concentration of potassiums inside. We had our salty part of our salty banana being the sodium chloride, so we get a high concentration of chloride outside the cell. We added on the fact that we have a lot of negatively charged amino acids inside the cell and calcium as a positively charged ion outside the cell. So here is the picture that we just described. However, to take all of those ions into account gets very complicated very quickly. So in order to illustrate what happens with these ions to give us our membrane potential, we're going to strip it down and simplify it. We're going to remove all the ions except for the potassium. And then once we see what happens to the potassium, we'll extrapolate that concept towards the other ions to get a more full picture. So if we only look at the potassium, we see a very strong concentration gradient of potassium because of the actions of the sodium-potassium pump, remember. High concentration of potassium inside the cell, low concentration of potassium outside the cell. And we see these potassium leak channels. I love the way that those are described because the name tells you exactly what it is. They allow potassium to leak out of that membrane. Think about a leaky rowboat. The sodium-potassium pump acts to bail water out. Those holes in the boat allow that water to leak right back in. So we pumped potassium in, it's allowed to leak back out. And remember, if you don't recall diffusion, go back and watch our video on simple and facilitated diffusion to get an idea of how this functions, where we'll always see diffusion of a solute from a high concentration to a low, in this case, through these sodium leak channels. So as we see potassium leaving the cell, it creates another problem, or at least another scenario. Remember, we had all of those negatively charged amino acids inside the cell. And we know that positive and negative charges like to attract. So for every potassium that we see leaving the cell, it leaves behind now an unpaired anion, a negatively charged ion. So one potassium leaves behind a negatively charged ion. Another potassium leaves behind another. Another potassium leaves, we get another unpaired negatively charged ion left inside that cell. Until eventually, we wind up with this excess positive charge on the outside of the cell, excess negative charge on the inside of the cell. But now we go back to the phenomena of positive charges and negative charges attracting each other. Now that we have those positives that we're allowed to leak out, we're going to see an opposite force simply pulling them back in. That's what we call an electrochemical gradient. So we see potassiums leave the cell based on their concentration gradient, and then we see those same potassiums pulled back into the cell by those negatively charged anions that were left behind along its electrochemical gradient. So we see potassiums leave through one door and enter back in through another. And this movement starts to look like something that we should describe as a current now, giving us our membrane potential or membrane voltage across that plasma membrane. The point at which the force pushing those potassiums out along its concentration gradient is equal to the force pulling those potassiums back in by its electrochemical gradient. We're going to call that our equilibrium potential. And that's going to be the voltage caused by that single ion moving out and then back in again. And we can actually calculate the voltage exerted by any of those ions by the Nernst equation, where we have the log times the concentration gradient outside compared to the concentration gradient inside. And when you do that type of calculation, we will see that the equilibrium potential for potassium is around minus 92 millivolts. That is the voltage exerted by the movement of potassium in the model that we just had. Now, potassium is not the only ion present. We already said we have sodium, chloride, and calcium outside. We have our other negatively charged anions inside the cell. All of those ions will undergo a very similar movement, where they will diffuse along their concentration gradient and then get pulled back across the membrane by their own individual electrochemical gradients. So the membrane potential that we had for potassium of minus 92 millivolts, that is created only by the movements of potassium. When all these other ions undergo very similar phenomena, we wind up seeing an overall membrane voltage, an average, if you will, of the individual influences of all of those isolated ions. Once all is said and done, and we average all that out, we wind up seeing an overall membrane potential of approximately minus 70 millivolts in most cells. This is our overall membrane potential of a cell. The effects that we see with all of the individual ions going across the membrane by a concentration gradient, then getting pulled back by its electrochemical gradient. I want to thank you for watching. I hope it helped.